So in our last video, I introduced you to the idea of the chromosomal theory of inheritance based on Morgan and Bridges' experiments. And now we're ready to roll up our sleeves and think about a new idea called linkage analysis. And I'm going to break this process up into two separate videos because I often find that this is a challenging set of ideas conceptually, and so it can be kind of helpful to break these ideas apart and be able to go back and revisit them in pieces while you're working through them. So in this first part, I'm going to introduce the idea of linkage analysis, and then in the second part, I'm going to take this idea further and kind of show you what can be done with it. Okay, so we're going to go back to Mendel. Well, sort of. So what we're going to do is we're now going to look at two traits in fruit flies. So we're going to cross wild-type fruit flies, and I'm going to indicate these as big P, big P, big E, big E. So this is my wild type. And so two dominant traits with mutants that have purple eyes and ebony bodies. So little p, little p, little e, little e. So here we have red eyes, symbolized by big P, purple eyes by little p, and quote-unquote normal bodies, so brown bodies by big E, or we can just call that wild type body, and little e is ebony body. Okay, so this looks a lot like a dihybrid type of situation, right? So if we do that cross, think about our gametes, right? Parent number 1 can only make one type of gamete, big P, big E. Parent number 2 can only make one type of gamete, little p, little e. So what that means is that our F1 generation is going to be 100% big P, big E, little e, right? Okay, so now for my next cross, what I'd like to do is I'm going to cross these heterozygous F1 individuals with a homozygous recessive individual. So again, this is one of these test crosses and back crosses. So big P, big E, little e crossed with little p, little e, little e. Okay, and so let's think about what we'd expect to get. And we could go through and draw out a Punnett square. I might not go quite that far, but we'll get close to it. So parent number 1 can make 4 types of gametes, right? So big P, big E, big P, little e, big E, little p, little e. And then parent number 2 can only produce one type of gamete, right? So little p, little e. So if we work this out, we've got big P, big E, little e. We've got big P, little e, little e. We've got little p, big E, little e. And then we've got little p, little e, little e, right? So now if we're thinking about phenotypes that can result from this, this individual is going to have the dominant phenotype for both of these genes, right? So wild type, wild type. This individual is going to be dominant recessive, then we've got recessive dominant, and then we've got recessive recessive. And for this cross, we expect to get an equal ratio of these, right? So 1 to 1 to 1 to 1 ratio. So if we're thinking about this with a total of 400 flies we count up, here's what we'd expect in the table here, is 100 of each type. All right, that seems simple. Well, that's all well and good, but if you do this cross, here's what you actually get. So I'm just going to write in a column here my expected numbers if I wind up collecting 400 offspring. And then next to that, I'm going to put what was actually found. So we get 180 that are wild type. We get 22 that have the wild type eyes and ebony body. We get 18 that have purple eyes and the wild type body. And then we get 180 that have purple eyes and ebony body. So what is going on here? Well, what I want you to notice is the original parental phenotypes way back up here. Those are showing up way more often than expected, right? So we expect only about 200 of those, and we got 360 total. And in contrast, these two mixed phenotypes, right? One trait wild type, one trait the recessive, right? Those show up much less than expected, right? So only 40 versus the 200 that we expected. So the conclusion we can draw from this observation is that these two genes, so the gene for eye color and the gene for body color, are on the same chromosome and they're linked, which means that they cannot independently assort during meiosis. So let's redraw this cross and how it looks if we actually track it on chromosomes through meiosis. So we started out with a parental generation that was big P, big P, big E, crossed with little p, little p, little e, little e. And so in parent number 1, if these genes are found on the same chromosome, what we will have is, imagine if this is metaphase 1 of meiosis. If this individual is homozygous dominant, there's going to be a location on each of these chromosomes that contains a big P allele. And there's going to be a location on each of these chromosomes that contains a big E allele. Okay, and then for parent number 2, for that same set of chromosomes, at that same location, there is going to be a little p allele, right? And at the start of meiosis, there will actually be 4 copies of that little p allele and a little e allele. So these cells are going to go through meiosis, and they're going to each produce types of gametes. So one parent will wind up with a gamete at the end of meiosis that has a big P and big E on it. And the other parent is going to end up with a gamete at the end of meiosis that has a little p and a little e on it. Then fertilization is going to happen, and the result, if we look at the start of meiosis in the offspring, so here's our F1 generation. Now what we have is offspring that have a big P and big E on one chromosome. And remember, these are sister chromatids, so they're identical to each other. And a little p and a little e on the other chromosome. So when this individual goes through meiosis and produces gametes, it's going to produce, we'll draw out all 4 gametes here. It's going to produce gametes that have big P, big E, and it's going to produce gametes that have little p, little e. Right? So if I cross this individual with another one that is little p, little p, little e, those gametes will all be 100% little p, little e. And so now if we look at all those possible combinations, you should see the same outcome, or that you should see that there's only really two outcomes here, right? So the offspring will either be big P, big E, little e, or little p, little p, little e, based on this idea that these two genes are linked and found on the same chromosome. This might lead you to a question. So if these two genes for eye color and body color are on the same chromosome, then how is it that we are seeing any of these individuals that have a mixture that are different from the parents? Aren't those impossible? And the answer is they are not, because of crossing over during meiosis. So it's possible to get a recombination due to meiosis. So we need to take a look at this with a diagram of some chromosomes to help make sense of this. So what these two chromosomes here represent are the chromosomes of these F1 individuals. So big P, big E, little e, right here. Okay? So if we think back to this parental generation, and we think about parent number 1 here, and what happens during meiosis, right? They're passing on a full chromosome that contains the big P and the big E. And let's just say this is the mother in this case, right? And then meanwhile, we've got another chromosome that's getting passed on that contains little p, little e. And let's just consider that to be the paternal chromosome. So overall, if these chromosomes stay the way they are and they separate during meiosis, what that's going to give us is a gamete that has big P, big E, and a gamete that has little p, little e. But if crossing over is possible, then we might also wind up with recombined chromosomes, right? So we refer to these outer cases as being the parental combinations, and then these inner cases as recombinants. And we can use the type of backcross that I'm showing you here to calculate the distance between two genes on a chromosome. So in this little cartoon here, I'm trying to show you the same thing that I just illustrated through the other chromosomes, right? So if I cross big P, big E with little p, little e, then my F1s have chromosomes that look like this cartoon here. So I have big P, big E chromosomes, little p, little e chromosomes. So if we picture meiosis happening, we expect to get mostly big P, big E together, little p, little e together. And so then if we're crossing these against little p, little e, and in color, then I'm trying to show you these combinations across the offspring that are produced, right? So here's big P with big E. Here's big P with little e. Here's little p with big E, and then little p with little e from that heterozygote parent. Okay? So from here, our total number of parental types, again, so is 180 plus 180. So our parentals, we got 360 parentals total, right? And for our recombinants, we've got a total of 40 recombinants here, okay? So the recombination rate is then the total number of recombinants divided by the overall number of offspring. So we have 400 offspring total. So our recombinants are 40 out of 400 offspring. So this gives us a recombination rate of, or recombination frequency of 0.10, or we could say, 10%, or we actually use the units centimorgans, which are equivalent to percentage. So 10 centimorgans is our distance between the P gene and the E gene. Okay, so now hypothetically, to put this in perspective, let's think about what we would get if two genes were unlinked. And that's actually the case in this expected column here, right? If these genes are unlinked, so if they assort independently, then we expect an equal ratio of all these different types of offspring, right? So then we can't really call anything parental or recombinant, but we can pretend that they're parental and recombinant. Then, we're going to get 200 for our parental types, quote unquote, 200 for our recombinant types. And if you divide 200 divided by 400, then you get a result that's 0.5 or 50 centimorgans. So the highest number linkage number you can get is 0.5 or 50, right? So if genes are very, very close together, you'll get a very small recombination rate. If they're very far apart, or if they're on different chromosomes, you will get a very high recombination rate. That's a lot to think about. So we're going to stop there for this video, and we're going to continue with this idea in the next video.